This is the basic structure of DNA. It consists of two strands forming a double helix running in opposite parallel directions. You can see with the nitrogenous bases that guanine is paired with cytosine and adenine is paired with thymine. We will demonstrate how DNA is replicated using colored Play-Doh. This is the overall structure we plan to represent DNA replication. Throughout the video, we'll go through each substance to demonstrate what its job is. First substance is known as helicase. It is known for unpacking or unzipping DNA. What it does is it separates the strands. Now, this doesn't seem too important, but it's vital for all living organisms. Helicase is an enzyme. The next substance is primase. Primase is RNA, and it is also an enzyme. It's important because it sets a primer for the rest of the DNA to be copied. Polymerase is another enzyme vital to DNA replication. Its job is to read the unzipped DNA from helicase so that it can make a pair for the DNA strand, adenine to thymine, guanine to cytosine. Single-strand binding proteins, sometimes referred to as SSBP, are the substances that allow DNA strands to be separated. Since they are held together from hydrogen bonds, it is very difficult for DNA not to be pulled back together. SSBPs are what make that possible. As mentioned earlier, DNA's structure is a double helix. The double helix consists of two strands, one being continuous, the other not. The continuous strand is known as the leading strand, while the other is known as the lagging. In the lagging strand of DNA, gaps are formed, referred to as Okazaki fragments. It is when these spaces form that the enzyme ligase comes into play. Its job is to replace the gaps caused by Okazaki fragments. With all of these substances, you have DNA replication, which is the process in which DNA is copied.